Okay, so now what we're going to talk about is a very similar looking process with some significant differences here. So what we're going to talk about now is meiosis. And so if you think about this, should make sense, but if you didn't have meiosis and you had a sperm and you had an egg, and the sperm and the egg both were like all your other cells, they would have each, in a human, 46 chromosomes. So when the sperm and the egg got together, you would end up with 92 chromosomes. And you might think that sounds good to have an extra set of chromosomes, extra 2 sets of chromosomes, but it turns out it's not good. Most things do not do very well. Either missing chromosomes or having extra chromosomes has significant consequences depending on the chromosome. Especially a whole set ends up being very bad, especially in animals. Okay, so what meiosis does basically is it takes your diploid cell typically, okay, and so this occurs in humans. We'll use humans as our typical model, but you should realize this can be true of plants and also true in other animals with some slight modifications depending on like the number of chromosomes and so forth. But essentially, like in the case of humans, males have testes, and in the testes there are diploid cells that will go through this process of meiosis and copy the information and end up making a haploid cell. In this case it will be a sperm, which has 1 set of chromosomes in it. It has an N value, and the N value is 23. In a female has ovaries, and in the ovary what happens is you have the same process, goes through meiosis, but in this case it's going to make an egg, and the egg has 1 set of chromosomes in it as well. In the case of a human, it's going to be 23. So the sperm and the egg then get together during fertilization and form this 1 cell, which is now a diploid cell called a zygote. So you the student, started off as this 1 cell. Your mom and your dad, this might be disturbing, but it's somewhat true, through the testes and the ovaries, made sperm and eggs and fertilized during sexual reproduction. The sperm fertilized the egg or the ovum, and you ended up as this 1 cell, this zygote. This zygote then goes through that 1 cell, starts off as 1 cell, then goes through mitosis, where you take a diploid cell, you copy everything in it, and you end up with 2 cells, then 4, then 8. And so you started off with this 1 cell, and then by going through mitosis over and over again, you end up with you as the individual after many replications of that. So let's talk about meiosis then, and how you make those sex cells, how you're going to make the sperm and the egg. And you'll notice that the similar steps to what we saw in mitosis, but in meiosis we're going to do everything sort of twice, and the first set of things is where all the big differences are. So first we'll start with interphase. So the cell goes through interphase in a similar way that the cell in mitosis went through interphase. So you go through G phase, S phase, G2, and you copy the stuff, and you have 2 sets of chromosomes and so forth, and there's your chromatin. So interphase is basically looks the same for the most part for our purposes. Okay? Then we go through a prophase, and you notice that we have prophase 1, metaphase 1, anaphase 1, because during meiosis we're going to go through everything twice. So we have a prophase 1 and a prophase 2. So in prophase 1, this is quite different than what happened in mitosis. So what's going to happen in prophase 1 is the nuclear envelope's going to dissolve, and your sister chromatids, remember they're connected, your chromosomes are going to pair up with each other. So you know that you have 2 chromosome 1s, for example, 1 from mom and 1 from dad. Okay, so they're going to pair up with each other, but this is what's really significantly different. They're going to form these structures called tetrads. A tetrad is where you have two homologous chromosomes with each having their two chromatids. So you end up with 4 chromatids there together. And then what's going to happen is at very organized spots and places, the chromosomes are going to break apart and swap places with each other. And that process is called crossing over. And that occurs in prophase 1 of meiosis. And that's significantly different than what happened in mitosis. Mitosis has nothing occurring like that. So what happens if I jump ahead really quick here, here's say 2 chromosomes, one chromosome 1 and the other chromosome 1. They're homologous because they carry the same type of information in the same relative area. They're going to form these structures called tetrads, and then they're going to swap parts. Now in this picture they just show you swapping the ends, but it can swap in many different places. So what ends up happening, you notice, is that this chromosome 1 is now not the same as this chromosome 1 was because it's got some of the blue part on it. So they're not really red or blue, but instead what they do is they have slightly genetic differences usually at some of these points. And so what happens is during crossover, you essentially are not getting a chromosome 1 that's exactly like your dad's or your mom's, but instead you're getting a chromosome 1 that's like a mixture of your dad's chromosome 1s. Okay? Because you know you're only going to get one chromosome 1 from your dad, and you're going to get one from mom. But instead of getting one exactly like your dad's, you're actually getting some of the genes from both of your grandparents, because your dad's 2 chromosome 1s and your mom's, instead of getting just one of those, because they've swapped parts, if you get this chromosome 1 or this one, you're going to get different information than is exactly the same found in any one of your parents' chromosome 1s. And again, that's different, and that does not occur in mitosis. So that's a significant difference in meiosis. Okay? During metaphase 1, the homologous chromosomes line up in pairs. That's also different. Okay, so instead of them all lining up like they did in meiosis they line up in pairs. So that in an anaphase, what happens is the homologous pairs separate, but the sister chromatids remain attached. Okay, and so then the cell goes through telophase, and then telophase 1 and cytokinesis. And what happens at that point, you'll notice that the cell divides, and what you have is you have your cell with your chromosomes in it, but the chromosomes are still duplicated chromosomes, but they're haploid at that point, meaning they only have one of the chromosome 1s and one of the chromosome 2s and one of the chromosome 3s, depending on the cell. In this particular case, in this picture, they're showing you that there's 3 in a set. In this, if this was a human, there would be 23 of these structures, and they'd be duplicated. Okay, so then from telophase 1 and cytokinesis, we go through meiosis 2. Meiosis 2 is very much like mitosis, but it's occurring now on haploid cells. So what ends up happening, you see the spindle fibers form, you see the chromosomes. In metaphase 2, all the chromosomes line up in the middle. During anaphase 2, the sister chromatids separate and become daughter chromosomes. And then in telophase 2 and cytokinesis, they divide and you end up with 4 haploid daughter cells. In the case of sperm, these each become individual sperm. In the case of the egg, it turns out really only one of these will become an egg, and the other 3 will be what we call polar bodies, and they'll just be pieces and sections of DNA that the cell ends up getting rid of just to reduce the chromosome number. So again, if we look at and compare the differences between mitosis and meiosis, in mitosis what happens is during prophase we can see the duplicated chromosomes, but in meiosis we get this crossing over event where you get genetic recombination because the tetrads actually swap parts. Okay? During metaphase of mitosis, all the chromosomes line up. During metaphase 1 of meiosis, they line up in pairs. During anaphase of mitosis, the sister chromatids separate and you end up with 2 diploid cells. During anaphase 1, what ends up happening is the sister chromatids remain connected and they separate, but they separate while they're connected, meaning that you end up with a duplicated chromosome in separate spots, but they still remain attached. So the main goal at the end of meiosis is basically this. You start with a diploid cell, you copy everything in that diploid cell, and then you divide it once, and then you divide it twice, and you end up with, at the end, 4 cells, and each of those is a haploid cell.